Alright, in this recording we're going to start moving into the next section of the course, and that deals with genetics and evolution. And we're not leaving behind the information that we've learned so far. You're going to see very, very acutely that we need to draw on what we've learned so far to understand these concepts, which are essential to biology. And so the first idea that we need to talk about is the idea of ploidy. So ploidy basically refers to the number of copies of the blueprint that we find in your DNA. And up till this point, we've referred to human chromosome number as 46, that you have 46 chromosomes, which is absolutely true. You have 46 chromosomes. Every human has 46 chromosomes. It's the number of chromosomes we have as a species. There are some exceptions with genetic diseases. But chromosome number is species specific. So different types of organisms have different numbers of chromosomes. But the idea of ploidy then can be applied to that chromosome number. And if you look at human karyotypes, these are pictures of all our chromosomes, you can actually see that you can pair up chromosomes. And so what you'll often hear then is that humans have 23 pairs of chromosomes. And you can see here that in fact each pair of chromosomes lines up, and two chromosomes look like each other. There's chromosomes in pair 1, 2, all the way through pair 22. Those are called the autosomes, and the sex chromosomes are the X chromosomes. So this is a female karyotype, and she is truly 23 pairs, whereas a male has 22 autosomal pairs and then has an X and a Y chromosome, which are not homologous. They're not a pair of chromosomes. So this is a very important concept. Working with this concept, and as we work more in genetics, it's going to become clearer to you, but we need to get it kind of out there now. Now, when you have two pairs of chromosomes, it's called diploid. D-I-P-L-O-I-D. Di for two copies. Right? If you are an organism that has but a single copy of every chromosome, you're haploid. There's also tetraploid, hexaploid, octaploid. So if you're tetraploid, you have 4 copies of every chromosome. There's lots and lots of plants that are tetraploid. Now, don't think that because you have a copy of a given chromosome that you don't need that chromosome. You need both of your chromosome 1. If you were to be missing one, things would be very, very bad. Probably wouldn't grow at all. It doesn't work out as being human. You need all 46 chromosomes to make you. It just so happens that those chromosomes are paired, that redundancy is necessary to build the organism. Now, what we're going to move into as we move into this idea of ploidy, it brings into the idea of sexual reproduction. And so when we think about having a pair of chromosomes, where did this pair come from? Well, in fact, one of each of those pair came from your father, and the other one came from your mother. And so those were handed off in what we call gametes. And we'll talk about this in just a second. But those two gametes combine to form a progeny. So you have a blue gamete and a yellow gamete here that comes together into the more opaque green progeny, this idea of ploidy. But both copies are necessary. Now, when we look at those two different versions, or those two different chromosomes that are in the pair, they're copies of those chromosomes. All the genes that are on those chromosomes are the same. But the versions of the different genes, the versions of the genes at those different locations on the pair of chromosomes may be different. This is the best picture for this, right? So you've got two chromosomes. They are copies of those chromosomes, right? They're in the pair. They're what we call homologous chromosomes, right? So they're pretty much the same, except for the fact that they have what's called an allele. An allele is different versions of a gene. So at this particular location on these chromosomes, there is a gene that encodes for flower color. So we call that the locus. So we're looking at two loci here. This is the locus for flower color gene. And in one of the chromosomes, you have the allele for purple flowers. It means you have the version of the gene that encodes for purple flowers. It's a gene that encodes for flower color. On the other chromosome, you have the allele or the version of the gene that encodes for white flowers. It's the gene that encodes a protein for flower color, but it's slightly different because it's white and not purple. So these homologous chromosomes are the same. They have the same genes on them, but they may have slightly different versions of those genes, which means they're not perfectly identical. You got one from your mom, one from your dad. And this is going to be the heart of how genetics works. And so we need to lay down this idea of ploidy and homologous chromosomes before we can understand genetics. So what we're going to do now is look at the sexual life cycle, or sexual reproduction, or what's called the gametic life cycle, and break down kind of some of the key terms to understanding then how genetics work. So if we look at the two adults in the lower left, they are diploid organisms. So they have 2 copies of every chromosome, or you could say 2 copies of every gene, but their total number of chromosomes is 46. If you don't have 46 chromosomes, it doesn't work, or it's just not right. Now, if you look at an adult human, there are 2 major types of cells. There's somatic cells and germline cells. We talked about these briefly before when we talked about mitosis. Now, meiotic division produces an exact copy of a cell. That's what we studied. And if you don't understand mitosis, if you don't have a very good grasp on it, now is a good time to stop and go back and review. Because if you don't have a good grasp on mitosis, understanding meiosis, the process of producing gametes, or cells for sexual reproduction, will be very difficult to grasp. If you have mitosis down well, the modifications that we see in meiosis will be easier to understand. Now, somatic cells are the body cells that you have. They have 46 chromosomes, right? Now, some of the cells in your body are not somatic cells. They're called germline cells. They also have 46 chromosomes. These are populations of cells that are maintained in the ovary and testes in the gonads. And the purpose of these cells is to undergo the formation of gametes. So they're going to go through a special type of cell division that only happens to germline cells called meiosis. And meiosis is going to have the ploidy. So if you are a 2N or a diploid organism, your gametes will be 1N or haploid. So sperm and egg are haploid cells. When they fuse, 1 plus 1 equals 2. And so what this does is it avoids the complication of doubling the chromosome number when you exchange or share genetic material with another individual of your species. So were I to combine my genetic material with another individual without having my chromosome number first, then your progeny or your children will have twice the number of chromosomes that you do. And if they go on to have children, then they will double that number again and again and again, and that is unsustainable. So there had to evolve some mechanism to have the amount of DNA that you're literally handing off to the next generation, so you can give half to your child and another individual gives the other half. So the chromosome number is maintained from generation to generation. Sexual reproduction allows you to combine genetic material between two different individuals to maintain and increase genetic diversity. And we're going to see that that is fundamental for evolution. Okay, it's very, very good for the species to have lots and lots of genetic diversity. So we'll come to that later, but this is the foundation of this. So these are some really big ideas in biology, and they're really important to understand. So this human life cycle, sexual reproduction, gametic life cycle, we go from a diploid organism that has some cells that undergo meiosis to produce gametes, these specialized cells for reproduction, right? That then fuse together to form what's called a zygote. That single cell that is now diploid again and goes through meiotic division to grow up into you or whatever organism it is. So the fancy word for that sperm and egg that come together is called syngamy. And of course, the word zygote is going to be super important. So we're going to see that almost all the eukaryotic life that we know has some form of sexual reproduction, and that a zygote forms somewhere in there. And that single cell then grows into the trillions of cells that make up most multicellular organisms. Now, again, this idea of a gametic life cycle is common to most animals, but it's not the only way to do this. So what you're going to see is called the sporic life cycle down at the bottom, which is most plants. We also call that the alternation of generations. We'll visit that later when we look at the plant kingdom. And then you have what's called the zygotic life cycle for fungi and some algae, which gets very unusual and very weird. And we're not going to spend too much time with that this quarter. But once again, realize that the way that we do it isn't the only way to do things in the world. Now, what we want to do now is spend a little bit of time talking about this process of meiosis. How do I have the ploidy for my gametes? What kind of cell division? How is the cell division different than mitosis? So what we want to review is this idea of chromosomes. So one chromosome on the top goes through duplication during the cell cycle to make a copy of itself. Right? Now this is an actual exact copy. I'm copying the DNA. This isn't about homologous chromosomes. I'm copying that DNA, and those two sister chromatids are attached to each other. This is one chromosome that is duplicated now in the middle. That will eventually split to form two daughter chromosomes, each distributed to the daughter cells. That was what we talked about when we talked about mitosis. We're going to see similar effects here in meiosis, and I want to use the same terminology. One chromosome, one duplicated chromosome, splits up into two chromosomes. Now, meiosis is about having the ploidy. So when I look at the top cell, the parent cell, you see it has two chromosomes in this example. Those chromosomes are going to duplicate themselves. Now you see the X shape of those chromosomes with two sister chromatids. But now when it goes to the first cell division that we see in meiosis, they separate from each other. And now we've gone from two chromosomes in the cell to one chromosome in the cell. That one chromosome will go through another round of cell division and split the duplicated chromosome into two now daughter chromosomes. So meiosis is a process of making gametes and having the ploidy. It's going to go through two rounds of cell division to do that. Right? So let's look at this, and we're going to see some familiar phrases. So prophase, metaphase, anaphase, and we'll see telophase on the next slide. PMAT still works. But you'll also notice, and I want you to pay very close attention to this, is the Roman numeral after each of those words. So prophase 1 is referring to a stage in meiosis. Prophase, with no Roman numeral, refers to mitotic prophase. And that's what I'm going to hold you to. So be very, very careful when you write these things down for answers, whether you're talking about prophase or prophase 1 or ultimately prophase 2, which we'll talk about in a second. Now, just like mitosis, we're going to front load much of the activity, the important stuff of meiosis at the beginning. So, if we're thinking about a germline cell getting ready to make sperm or eggs, it's going to undergo meiosis. So here's the cell on the far left that is exiting G2 and going to start the M phase of meiosis. All right? So we're going to see breakdown of the nuclear envelope. We're going to see condensation of the chromosomes. We're going to see the formation of the meiotic spindle. That's just like prophase in mitosis. Some additional things that are going to happen here is that chromosomes are going to find their homologous partner. So that means that if it was human, if this was a picture of a human meiotic division, you would see 23 red and blue pairs lining up with each other. And you can see now as they find their homologous partner, they do an event that's called crossing over. And so you see that fancy word pointing to little circles at the crossing over point called a chiasmata. And that literally is where the DNA of the chromosome that you got from your mom crosses over with the chromosome you got from your dad and splits, and they exchange those parts. Truly mixing up the DNA, right? So the same genes are all there, but the versions of those genes that may be on the different chromosomes is now mixed up. Okay? Because what you're literally doing when you're going to hand off each one of those chromosomes to your progeny is it's a mix of the chromosomes that you had. And that's what happens in prophase 1. So prophase 1, we see tetrad formation. We call it a tetrad because tetra is 4. And there are 4 chromatids now that are doing this crossing over event. Right? We see crossing over, an exchange of pieces of chromosomes in the homologous pair. This is increasing genetic diversity. So from prophase 1, we go to metaphase 1. Metaphase 1 is very similar to metaphase, but instead of all the chromosomes lining up on the metaphase plate, they line up in pairs. And this is key to having the ploidy. So the tetrads line up instead of the individual chromosomes. And if that sounds confusing to you, go back and look at mitosis. Notice the details. In mitosis, the chromosomes line up on the metaphase plate. In meiosis, the tetrads, or the chromosome pairs, line up on the metaphase plate. And when they line up on the metaphase plate, they can get separated, not separating chromatids, but separating the pairs. So you can see here in this picture now, anaphase 1, one chromosome is headed towards the top of the cell, and one chromosome is headed towards the bottom of the cell from each pair. So 2 chromosomes are headed north, 2 chromosomes are headed south. I started with a cell with 4 chromosomes. Ultimately, what we're going to see is a cell forming that has only 2 chromosomes. Now, granted, each of those chromosomes is duplicated, but it's only 2 chromosomes. I've halved the ploidy. Now, that cell will immediately go into meiosis 2, the second round of cell division. Now I only have 2 chromosomes. So prophase 2 is going to be a breakdown of the nuclear envelope, if the nuclear envelope reformed at all, another formation of the mitotic spindle. Now the chromosomes are going to line up on the metaphase plate, and this is going to look just like mitosis, but with half the number of chromosomes. Line up in metaphase 2, pull the chromosome apart, the 2 sister chromatids apart in anaphase 2, and then we split the cell in cytokinesis once again. And now we have 4 cells, where we started with 1. Each cell has only 2 chromosomes, where the original cell had 4. So this is a great slide to study, because what you're doing here is you're comparing mitosis and meiosis all in one slide. So look at the parent cell. It has 4 chromosomes. It is diploid. That 2N means it's diploid, right? In mitosis, on the left, you see exact copies of the parent cell as a result. Both are 2N. Both are diploid. Both have 4 chromosomes, 2 blue, 2 red. In meiosis, you're going to go through all the steps that we talked about, meiosis 1 and meiosis 2, and the daughter cells now, you have 4 daughter cells. These are going to be gametes. Each has not 4 chromosomes, but only 2. And because of crossing over, each is genetically unique. So I have a daughter cell that is red, and another chromosome that's red with a little bit of blue. And then I have the other chromosome with a little bit of blue and an all red chromosome, and so on and so forth, right? So this then results in unique combinations of alleles that we didn't see before. Okay? So when we look then at this process in greater detail, and we're going to need this as we get into genetics, so we'll come back to this idea, is that when we zero in on metaphase 1, when tetrads are lining up on the metaphase plate, the way they line up on the metaphase plate is random. All right? And so this is called the law of independent assortment. And so this idea that even without crossing over, that these chromosomes, the one you got from your mom and the one from your dad, could line up randomly on either side of the metaphase plate, can result in different genetics in the gametes. Right? It's combination 1, combination 2, combination 3, combination 4, and that's just because they're of the random alignment on the metaphase plate. Okay? You throw in crossing over on top of that, and again, the whole point here is that you're really mixing it up. These pictures are simple, right? And they may not seem simple to you, but you will study them. By the time we're done studying this, it'll be a simpler process. But now, blow this up and take it to 23 pairs of chromosomes. That's a whole lot of shuffling going on, right? And so that really ensures that we try to maintain genetic diversity, and we're going to talk about that more as we move into genetics.